and we decided to do our presentation on the discovery of DNA. There are three important people who play a significant role in the discovery of DNA and the double helix. They are Johann Friedrich Meissner, who discovered nucleic acids, in which Christine and Melissa will be talking about, and Watson and Crick, who proposed the double helix structure of the DNA molecule, in which Dr. will be talking about. But before I leave, I would like to share a poem we call DNA. DNA, or deoxyribonucleic acid, a mouthful, which should be a poem. Adenine, cytosine, guanine, thymine, which should be the names of angels. Creative bond of adenine with thymine, cytosine with guanine, A to C, C to G, which is love as chemistry. In just 4 letters, clustered into 3. 1.8 meters of DNA in each of our cells, sparkling spiral strings, silver threads, wound into structures less than 1/10 of a millimeter across, leaving plenty of room on the head of a pen for angels. Invisibly clustering the space of a seed, reaching to the sun and back 600 times, bundled into 23 chromosomes, paired 3 billion letters planted with genes, nuggets of DNA in encrypted verses. Chemical factories with magic and dancing at the heart, realized script called to life, creating proteins, amino acids, which are the shared spoken word of skin and wing, peacock and goat expression, mechanism of holy poker, which has written itself out of nothing. The original miracle found among stars, writing deducted life deduced from the blindness of existence. Evidence and imagination beyond bone, molecule of bone to the dream of bone. Maurice Wilkins, Rosalind Franklin's X-ray crystallography, which sounds like it looks for the hearts of breeze. Shining X-rays through the DNA molecule, catching the scattered pattern. James Watson and Francis Crick dreaming the double helix, orderly flux, the dancing spiral, alive with love and creativity. Artist shape that is neither life nor chemistry, idea or dream, but all, synthesis for imagining. Picture for understanding, practicality as E equals MC squared is expression. So a ladder, elegant and twisting with something of a swan's neck. Backbone poles of alternating sugar and phosphate groups, attached bases forming rungs at each waist twist. Loyal partnering of bases informed by Chargas rules. So Watson and Crick suggest each rung was composed of a pair of bases, joined by hydrogen bonds, shackles, A always forming bonds with T, C always forming bonds with G. We've discovered the secret of life, Shepherd classes. And here's Christine and Melissa.
So the first discovery of DNA. The date February 26, 1869 is an important one where the start is run. It starts in Tübingen, Germany with a Swiss physician. His name was Friedrich Meissner and doing research was his mission. Following in the footsteps to practice medicine like his father, but due to a medical hearing loss, he didn't even bother. He graduated from the University of Basel at the age of 23, but decided to study biochemistry with his MD. Under the guidance of Adolf Strecker and Felix Hochseiler, Meissner worked in a lab. It was a converted laundry room and kitchen, and the surroundings were drab. Meissner wanted to determine the chemical composition of cells. He decided on lymphocytes as the article cells. Hochseiler had worked with hemoglobin for years. He put a lot of work into it, all of his blood, sweat, and tears. He suggested to Meissner to stick to the plan, but use leukocytes, and with that idea he ran. Meissner gathered pus from wounds at a hospital in town. He collected all of this without a bit fuss or frown. He obtained the leukocytes from the tissue, but choosing a salt solution was the issue. After washing the cells, he looked under his scope. Categorizing the proteins and lipids was his ultimate hope. In this day and age, there were too many proteins to classify. so Miser started to look for an alternative method to go by. During his work, he found a substance that was odd. Little did he know that it was the molecule of life that controlled the development of your body. He observed that lowering the pH caused the precipitate to turn white, but with an alkaline solution, the substance disappeared from sight. He concluded that proteins were no longer there. Miser couldn't stain it yellow with iodine, that of which he was aware. So what was this substance and what was its fate? It wasn't a protein, lipid, or carbohydrate.
It resided in the nucleus, all the experiments did show. The function of this organelle was something scientists did not know. Although this organelle was discovered as early as 1802, the concept of its existence was fairly new. With people not knowing and scientists in the dark, in 1866, Ernest Haeckel found a way to make his mark. Haeckel proposed that the nucleus contained the hereditary trait. From that, Miser developed protocols to get the nuclei in an isolated state. To break apart the cell, Miser used pepsin from the gut of a pig. Isolating the enzyme was difficult, but he definitely didn't dig. Miser washed the cells with hydrochloric acid and cold temperatures that were hard to fathom in order to break open the cell membrane and drain the cytoplasm. When he added water and ether, the lipids went away, but the nuclei that fell to the bottom of the vessel was here to stay. Miser decided that nuclein was its name, a term still preserved today. Deoxyribonucleic acid is one and the same. The nuclein contained phosphorus, which he didn't expect, carbon, hydrogen, oxygen, and nitrogen, but no sulfur that he could detect. Miser couldn't believe DNA was responsible for all embryonic information. It was stored in the form of stereochemical carbon conformation. With the number of stereoisomers nearing 1 trillion, Miser concluded that the encoded hereditary information was enough for the life forms of millions. If there was an error in a molecule, it wouldn't have an effect. The discovery of intact alleles would compensate for an effect. Miser walked away from his research to study salmon instead. Therefore, the study of DNA could not get ahead. It took 100 years before Watson and Crick discovered its structure and function to give us the knowledge today of DNA and protein production.
I'm just going to give you a little background information. Francis Crick, he was born June 8, 1916 in Northampton, England. He began doing science experiments when he was just 10 years old. And he graduated with a degree in physics from the University College in London. James Watson, who was born April 6, 1928 in Chicago, and he enrolled in Chicago University at 15. He majored in zoology, and he received his PhD in genetics from Indiana University at 22. He's only 23 when his greatest discovery was made. Maurice Wilkins was a New Zealand-born but a Cambridge-educated physicist. He worked with Rosalind Franklin, who was the only one of the 4 scientists that had a degree in chemistry from Cambridge University. She was an expert of X-ray crystallography, and she worked with Maurice Wilkins, so they both did. Watson met Wilkins, and they looked at the X-ray photos of DNA, and then Watson was hooked. Crick, a physicist in biology, was writing a dissertation on X-ray crystallography. When he met Watson, who needed a partner or colleague, if you may, for work on the structure of DNA. Watson and Crick, who put together several models of DNA, came across Franklin's X-ray photographs in a most deceitful way. The 4 scientists announced the structure of DNA, and the structure is the one that you see before you today. The Nobel Prize was awarded to Watson, Crick, and Wilkins for the determination of the structure of DNA in 1953. Unfortunately, the prize isn't awarded posthumously. So the dearly departed Franklin, who succumbed to cancer just 4 years prior, did not receive the prize for what her work so greatly inspired. Thank you for your time.